Hi guys, uh, thank you for uh, checking in to review this material. So this one is about chromosome variation. So let me start share the screen here. Let's see, share. Okay. All right. So you can see this PowerPoint slides right here. So we talk a lot about uh, the genetic stuff, and we talk about the general like gene structure, DNA structure, chromosome structure, right? And with this one, I like to explore into something that's very interesting. This is about the chromosome variation. So with this one, um, I like to uh, provide the important knowledge about the variation among us, and to see that if we can use this variation to conduct some type of that DNA profiling. So in the end, we will talk about how can we use it to conduct the DNA profile, and this is very important to identify the individuals for the criminal analysis, and also the forensics. Right. So the material coming from the textbook, chapter 13 and 14, 13 talking about the chromosome. So we would use this material to describe these chromosome. And then we will talk about the constant allele frequencies. So this one is talking about more like statistics part, the probability. So one is more biology, the other one is a little bit like covering the math part of this portion. So I think this is good for those who are coming from the computer science major, and with this one, you kind of get the sense how we can analyze this big data into into several like mathematical model. All right, so that's that one. So first, this is chromosome. So this is a typical karyotype that's showing you beautiful, the 23 pairs chromosome from each human somatic cell. And so you can see that the very basic chromosome. So you got, for these somatic cells, you got a pair of the chromosome for each chromosome, because we got one from father, one from mother, and they may carry different alleles. They may carry some variation. What are those variations? Has ever occurred to you that why are we all look very different? We may have something in common, but we are all, in the end of the day, we are all human beings, right? But why are we all different? Is there a genetic reason for it? Yes, there is. Because even though that we are considered normal, but how can you quantify, how can you tell a person is normal if they are all different? Why do this call normal? The other one is called abnormal. How can you do that? Why do you call this one a mutation, but the other, all very different human beings, are considered wild type in a way that we are all normal, right? So what's that? We are all very different. So if you want to say that you are normal, you can also say you are abnormal, right? You are unique. It's maybe a better way to say it. You are unique. Everyone is unique. All right. So I want to. So what's that unique part? If we look into the chromosome, basically everyone has. If we just look into like zoom or looking at it, there are some variation, but overall they are relatively similar, right? But they are different. So that's why we are all very different. Um, so what's in this chromosome? In the chromosome, the structure or the shape of chromosome typically has two arms. One is short arm, we use the letter P. The other one is long arm. We use a letter Q. P represent petite, so it's small. And these two arms are connected with the centromere. So this is the centromere. So centromere, basically this region doesn't really provide a lot of protein encoding. However, this region is an important juncture between two arms, large arm, short arm, and the long arm. We can also use this long arm, short arm, and long arm to conduct some labeling. So a typical way to label gene or label a locus is to use PQ, and then we can further use the bending pattern in each arm, and say P. So here is the chromosome 7, and we can have P and the Q, and the Q will first will have the separate into different larger band. So we can have band 1 here, and then we sorry, what's that? So here is the larger region. So here is region 3, because 1 is usually from the center. So we count from the center. So the region 1, region 2, region 3. So here probably the region 1, all right? Region 1 here, region 2, and the region 3. So we can have chromosome 7, region 3. In the region 3, we can further divide it into the band 1 and then sub band. So band 1, band 2, right? And then here in band 1, we can have sub band 2. So that's how we quantify, we label a location of a gene. 7Q is in the long arm, 3, region 3, 1.2. So this is an example of the gene CFTR. So something like that. But from here, you can see that this is very much like typical chromosome. Short arm, long arms, and centromere. This one can also provide us mapping. So as you can see here, this mapping is also called an ideogram that provide us a schematic chromosome mapping. So you can have P and the Q centromere. So when we have this mapping, we start from the centromere. So centromere is typically like 1.1, 1.2, 1.1. So this is the region 1, and then we probably can, so for example, region 1 is this region, and then we have a region 2, and then from the region 2 we can have the band 1, region 2 in band 1. So this is band 1, this is band 2, something like that. Yeah. So that's that. Now, centromere is the region that doesn't really provide the coding for the protein. So this is not like functional segments of the chromosome. This is more like a structure support of the chromosome. Structure support means that it connects P and the Q connect two arms. All right. So that's that. So centromere connect the short arm and the long arm. This is the, like the centromere in the typical cell. However, we know that during cell division, this centromere also plays an important role in that division. Divide what? Divide two sister chromatids. So I'd like to show you this one, maybe better to show you this one here. So this is the uh, chromosome before the replication. You have one from mom, one from dad. So it's very much like this one here, one from mom, one from dad, right? And then after the S phase, we replicate this chromosome, duplicate this chromosome. Then each one will have the other one. And the other one, these two are called sister chromatids. Now these two are large part are all separated, but there is one juncture that these two are connected. This is the centromere. So the centromere is the structure, provide this structure holding to hold large arm, small arm. This is also the region that two sister chromatids binds together. So that's this one here. So centromeres, after the chromosomes are duplicated in S phase, after that it generates sister chromatids. These two sister chromatids are connected by the centromere. All right. And one thing, there are two other things that I like to share about this centromere during the cell division is these two important, actually one very important protein complex. This is called the kinetochores. Kinetochore. That's right here. Kinetochore. These kinetochores are protein complex. These include more than 13 type of proteins, 19 actually, 19 type of the proteins. So it vary depending on the chromosome. Different type of chromosome may have a little bit variation of this uh, kinetochore. And this is an important functional proteins that provide the division of two sister chromatids. Because this is a protein complex, so it includes several proteins. We are not going to go into the detail about all these different proteins, but one protein's importance commonly known as the CENPA. This is the centromere protein A. This one is more or less important to control the cell division, sister chromatids division. This centromere protein A, CENPA, is a specialized histone. So we talk about this histone. Histone is a protein to hold the DNA together. So DNA is coiled and condensedly packed. So that proteins to hold the DNA together are histone. We also know that histone can be modified by methylation, etc. So this special histone located in the centromere is called histone H3. And this one, the specialized histone H3 is called the CMPA. So that is one important component in the kinetochore. So that's that. So this one just showing you a visual idea about the centromere and the kinetochore, that you can see that these two sister chromatids are about to separate. What's the phase is that one? We see this Since the chromatids are all aligned in the equator plate, what's that stage? What's this called? What's this phase is called? This phase is the metaphase, right? And the next step is the anaphase. So, so this drawing is not exactly accurate because these mitochondria actually connected with the centromere, connected with the kinetochore. So that's a better one. So I probably need to choose another figure. All right, I just replaced this figure. This one is more accurate because this spindle fiber, uh, in fact, connect with the uh, connect with the kinetochore. Kinetochore. Yeah, connect with this kinetochore. So that's the function of this kinetochore. They basically hold between the spindle fibers. These spindle fibers are the one of the cytoskeleton we mentioned very early in the probably the first week we talked about the cell. There are three types of cytoskeleton, and this one is microtubule. So, and that holds by kinetochore. Kinetochore holds on one part holds that microtubule, and the other one holds the centromere. So that's that. So that's the centromere. All right. And mm, the science to look into the chromosome structure is called the karyotype, and that is a typical one. That karyotype get a standing of this chromosome, and we can have in the old time we can identify different length of this chromosome, and then we can sort it, and we can identify these 46 chromosomes or 23 pairs of chromosomes. Nowadays, we can even do a different standing to stand different bending patterns of each chromosome. This is the typical human karyotype. This one is the chromosome after the S phase that the chromosomes are duplicated. So you can see that chromosome one, so we can kind of sort it. How do we sort it? We sort it based on the size. So from 1 to 22 is from the longer one to a shortest one. And then we have so this autosome. And then we have the sex chromosome, XX and YY right here. So that's that. So that's the karyotype. From here, we can kind of distinguish or classify the chromosome into 4 different categories, depending based on the length of this P arm and Q arms. So as you may see here, some of these chromosomes have almost equal P arm and Q arm, but some has very tiny P arm, right? So there are basically 4 different categories. We can have this metacentric chromosome, meaning P arm, Q arm are equal. We can have the submetacentric chromosome, meaning that P arm is shorter. Visibly, we can see it's shorter than the Q. We can have the acrocentric. This means that the P arm is much, much shorter. Then we have the heterocentric, means that P arm is invisible, barely visible. So that is different type. But I won't quiz you on this. I just need to know. You just need to be able to distinguish all these 4. But you just keep in mind that this one is one that we will study a lot. Um, this one is also include this five type of the chromosome: 13, 14, 15, 21, 22, and the Y. So that's right here. 13, 14, 15, 21, 22, and the Y. So this is acrocentric, that a lot of the chromosome translocation happens to occur in these five autosomal chromosomes. And so we will see this one more often. Just by the way, 21 is the one that when human have extra chromosome in 21, then the patients may develop the Down syndrome. That is the most common type of the trisomy, a type of the chromosome abnormality that we will also talk about today. In addition to that, this type of the acrocentric chromosome, you can see that there is a little bulb right here and with little connection. And here is right here. So this structure is called cellulite and stalks. So with this, say 21, this acrocentric, 5 acrocentric chromosome have this bulb-like ends. This is called the cellulite and this is called stalk. These stalks, even though that it's very look thin, this stalk provides the DNA to conduct the ribosomal RNA. And this region also collectively form the nucleolus inside of the nucleus. So that is an important information that you guys also need to know, that what are the ribosomal RNA is transcribed from. They are transcribed from the majority of those are transcribed from these acrocentric chromosome in the region of this stalk area, uh, in this short arm, in this stalk area. So that's that. One thing about this chromosome shape is that we need to have an idea that even though it may appear very beautifully, that as we always seen, if we look into the DNA structure or chromosome structure, we always have a very beautiful karyotype like this one here, right? That's very typical that we will get if we present with a chromosome. That's how we can appreciate the short arm, long arm, and the centromere, etc. But the one thing you need to keep in mind is that this shape is not always like this across the cell's life. So I want to go back to the very first lecture that we have in this course, the cell cycle. So that is the cell cycle right here. We can have the interphase G1, S, G2, and G0, and the mitosis, cell division. While we may see this X shape, and we see this X shape very typically during for the cell which is about to be divided. So we can have a condensed chromosome. We can appreciate the short arm, long arm, and the centromere. We may also see the cells already have the duplication with two sister chromatids, right? So that is in the cells which is about to get divided. However, this beautiful, easily recognizable X-shaped chromosome is not always the case. Uh, uh, so in particular, that in the interphase, that before the division, the chromosome may be appears more realistically like a ball of the spaghetti, like this one here. So this is basically still very tightly condensed chromosome or DNA, and this is the chromatin fiber. That's right here, the chromatin fibers before they got further, condensed. And this chromatin fiber, if we just opens up, we may see it like this, chromatin, like a ball of spaghetti. And if we further do a staining to identify different chromosome or chromatins from 1 to X, then we can see that they are, even though they are like a spaghetti, and they are kind of like a tangled together, but we can see that they have the major territory in the nucleus. And this is going to the term called the chromosome territories, like this one here. So it's it's good to know that we can appreciate the structure of the chromosome like this one here, or like this one here. However, it really depends on the phases. That in some phases, like interphases, that we may not be able to get a very condensed packed chromosome, but we rather got something like spaghetti, and we cannot just distinguish them based on their shape. We can distinguish based on the staining, and we can see that they are located in different in different territories. So that's that. So this one is difficult to analyze. So we will use this one to analyze, and that's and to describe you some of these chromosome shapes, chromosome variations. So this is the common one. This is from the textbook that we can see this is already sister chromatids connect with the centromere, and and we have short arm, long arm. There are some other things that we need to pay attention on. So we talk about the centromere, we talk about P arm, Q arm. One thing we need to pay attention is this chromosome may appear like dark density, light density, dark density, light density. What are the difference? Well, this difference represent the patterns of these DNA sequence. So in this dark sequence, this usually represent the region with a lot of repeats. Say that same sequence will repeat many times. So that is a repeat because they are repeated. So their package can be appear more condensed. And in this light region, this is the region that's not so many repeats. So this codes, this sequence here may contains more of this protein coded gene. Uh, so they are meaningful writing, they are not repeated sequence, okay? So that is this light region. This regions is called the heterochromatin. So this dark part, this is containing a lot of repeats. This one, as you can see, our gene, half of these chromosomes are heterochromatin. They carry non-coded repeats. And half of our chromosome are non-coded repeats. So they don't really get any proteins, but they are there. We also have this region. This is all euchromatin. This region contains the protein-coded gene. So that's that. All right. So that's these two region, euchromatin versus heterochromatin. The euchromatins are protein encoding sequence, and they don't appear very condensed because they are very, it's like writing, if for a meaningful writing, of course there is repeat, here and there, but you won't repeat it like a lot of the same words. It will be marked as a grammatic error, right? So that's that. And in contrast, these heterochromatins consist of these repeats. And these repeats, you need to know that is, even though they are not like coded gene, protein coding gene sequence, but they actually almost like take half of our genome in our body, in our DNA. So why do our nucleus need those? Why do we need space to carry those repeated genes, right? So that's something quite interesting to think about. So this could be a part of the quiz question if we, yeah, I think we have the quiz question, right? I think so. Yeah. So that was the difference between the euchromatin versus the heterochromatin. That one is the contains the repeats, the other one contains the protein encoding sequence. All right, so let's look at what are these repeats. Even though these are not proteins encoding sequence, but they are half of us, half of the chromosome. So what are they? Why are they? Why is a more difficult question to answer? But what are they is easier to answer. We observe researchers, look into the DNA and get a very good understanding. We already have very good understanding of these repeats. So the repeats, for example, like this repeats, ATTCG, ATTCG. Is that repeats? Yes. So you have this is a unit and it repeats 3 times. These are the sequence. They don't really provide a protein. Otherwise, you will have very strange proteins come from it, right? So this doesn't really provide the protein coding, but they are there. They are in the heterochromatin. They are almost half of the genome are repeats. And there are two types. One is called tandem repeats. Like this one, this repeats like appear right next to each other. The other one is called interspersed repeats. That is a repeats, but more like here, there, something like that. So that's two types. These tandem repeats are very important. Even though that's not protein encoding gene, but it's important in a way that you and me actually are different a lot in these repeats. The code itself, the sequence, is very universally the same. That I have this ATTCG located in chromosome, one of the chromosome, the same location, the same sequence. But the number of repeats vary between me and you. All right. So that's why we can use this one to conduct a DNA profiling. That this genes, this sequence repeats, varies, the numbers are different. It doesn't affect the function of our body because proteins is not protein encoded gene. They probably has the special function, really, depending on the location, but not fundamentally affects the human's function, right? But it's the length sequence is the same, but the length of that repeats vary between us. Of course, for people are biological related, their repeats number will be more or less similar. But for the people who are further away, the repeats number are different. So that's how we identify the DNA. That's how we identify, your offspring with some other people. We basically check their repeat length, and we can get, say, these repeats. We can get about, the typical one is say 10 different location, and to quantify the number of the repeats, and to see whether these 10s are all the same. If this all the same, it must be you. This DNA samples must come from you. If this is 99% similar or 90% similar, then this probably is from your relatives, your son, your father, your mother, etc. So that's that. So let's look into these tandem repeats. So something that we need to know, which is that the unit is the repeat unit. The length of these units can be one base pair. Is also called mononucleotide repeat. A typical one is A, right? And also could be several thousand base pair long. The length of that repeats, the number of repeats, is called it could be the number, is also called the variables. So it really depends on the way we say it, but the length is a typical way to say it. Number variable is also a common way to say it. It's individually different. And so that's how we can get this sequence, and to see that the length, whether these two are matched or not, similar or not. So here, okay, another thing is that these repeats, all together in that region is called a satellite DNA. If this repeats units ranging from 10 to 60, this satellite is called a mini satellite. These mini satellites are commonly seen in the centromere. So the centromere basically is it's comprised of these repeats, and a lot of these repeats is the mini satellites, meaning that the units is about 10 to 60 base length, and it has multiple repeats. If the repeats is less than 10 base pair, 1 to 10, then this is called the microsatellites or short tandem repeats. These short tandem repeats, I will just reduce this one here, because short tandem repeats can include both mini and micro. So I will just leave it like that. It's more consistent. And this one, microsatellites, can be found in the telomere. We didn't really talk about telomere yet, but we will talk about that. So it's here is the tip of the chromosome. So you can see this one is also a heterochromatin that basically consists of a lot of repeats. So that's that. All right, so that's here. So that's the mini satellites and the micro satellite. So here is some example of these repeats. Here are example of these microsatellites. So the repeats has these units, 1 to, the sequence amount is 1 to 10, right? So it's A, between these two, and this is mononucleotide repeats. This is dinucleotide repeats, CA. This is trinucleotide repeat, CAG, etc. And tetra 4, 5, 6. So that's that. These all are tandem repeats, meaning that these repeats are connected one next to the other. The number of the repeats are different between individuals. So this is the term called the variable number tandem repeats. VNTR. These VNTR are different between individuals. So we can use this to conduct a DNA profiling. Sequence is very much the same. Location is very much the same, but the lengths are different. So this is the DNA profiling. We will cover that portion here. Is to take the advantage of this variation VNTR to offer particular short tandem repeats. Short tandem repeats include the microsatellites and the mini satellites. So that's right here. That's why I removed that. So this is satellite DNA for this mini satellites and the microsatellites. There was there a delay? Sorry. Their sequence is the same across human being, but the lengths are different. And we can use that length, this length in the term is called the variable number tandem repeats, VNTR. So for example, the VNTR in two individual, like this is individual one, you can identify the beginning, that's the same. The ending, that's the same. But the sequence, that's the same. But the repeats, the number of the repeats are different between individuals. Individual ones have 5 repeats. Individual 2 have 7 repeats. So that could be different. VNTR in 2 different individuals. So you got the samples, cell samples, and you can compare it with the suspects and to compare their VNTR of these STR, short tandem repeats. And this is one STR. We can have how many STR? The FBI use 13 STR. So this is the chromosome, and use this autosomal chromosome. We now have very robust technique to cut these repeats. This is the repeats, and people with the length of the repeats can be varied. Okay, can be different. If all these 13 are exact the same, man, that is you. You know, that sample is from you. And if that is one difference, then it probably is from someone of your relatives. So that's that. All right, so that is repeats, and also telomere is on the tip and both tips of the chromosome. This also contains a lot of repeats. The repeats is micro repeats. The sequence is the TTAGGG. So this is also a quiz question that what is the sequence of these repeats in the telomere, TTAGGG, tag, in a way, right? TTAGGG, all right. So that's that. And the one thing about the telomere is that these repeats get shorter and shorter after cell division. And reach the point that you don't have the repeats, or it's too short, then the cells stopped divide. So by looking at this telomere length, we are able to identify the age of this cell. And the tumor cell is something different. Tumor cell is a mechanism to prevent this one shortening. So tumor cell can repeat many times while they are not going to stop repeating division. So that's that. Here is a collection of this key concept that we have talked so far. The chromosome structure, you need to know, you need to be able to identify the short arm, long arm, P arm, Q arm. You also need to be identify two major regions, centromere and the telomeres. You need to know that the centromeres hold these two arms. It's also an important region for the cell division. It's related to the protein complex called the kinetochore. And also the chromosomes, we have 23 pairs. And the scientific principle to sort these chromosomes based on their size is called the karyotype. And so.
I'm going to go to your school today.
Yes, yes.
Yippee hooray! Thanks.
All right. Yeah.
Thanks a lot.
All right. All right. So another important term is called the cytogenetics. So let me see. My kids just call me, and he probably has something to tell me. So I will. Okay, cytogenetics is one to talk about these chromosome variations. So I think I will just stop here for now, and I will make another video to continue in it. All right. So I will just stop here, stop recording.